Hello, everybody. I hope you're all having a great day. My name is Sciarratta, and I'm currently a sophomore biotechnology major. And through the Immersive Scholars Program, I was given the opportunity to conduct research within the Lee Lab. And my experiment focused on how the DAF12 nuclear hormone receptor impacts transgenerational longevity in these tiny microscopic animals called C. elegans that we use as our genetic model. So why is it important for us to study longevity and aging? So through longevity, we can compare the aging process of young individuals to older individuals. But what if we were able to add extra years onto our lives? To affect aging, we study chromatin, which is how the genome is packaged by histone proteins. And at the histone level, there are two distinctive marks that help manipulate longevity. The H3K4 active mark, which is added onto the histone tail by WDR5, and the H3K9 repressive mark. And in our WDR5 mutants, there is a lack of WDR5, which means that there is no H3K4 being added onto actively transcribed genes. And when this occurs, there is an accumulation of the repressive H3K9 mark within the germline over multiple generations, resulting in our WDR5 mutants being 30% longer lived than our wild type. And so because this was such an interesting effect, we wanted to understand how the accumulation of H3K9 within the germline impacted the soma. And so previous publications have stated that the WDR5 mutants may live longer because they have an increased activity in the lipid metabolism pathway located within the intestine. So how does something happen in the germline affect the entire body? There must be some line of communication between the two, but the mechanism that acts as the telephone is not quite definite yet. But one potential telephone mechanism that we have discovered is the DAF12 nuclear hormone receptor pathway. And because of this, it's known for its participation in the signaling pathways between the germline and the soma. And so we wanted to test if our transgenerational mutants could ever attain longevity if we remove this DAF12 nuclear hormone receptor pathway entirely. To test this, I measured the lifespan using our wild-type DAF12 single mutant strains and our WDR5 DAF12 double mutant strains. Our wild-type strains demonstrated a normal lifespan through the longevity assay. And additionally, our DAF12 mutants demonstrated a very similar trend in lifespan compared to wild-type, and there was no statistical significance between the two strains. However, in our double mutants, there was a statistical significance between the wild-type and the double mutants, and the WDR5 DAF12 double mutants were 26.67% longer lived compared to our wild type strain, which is like huge. And this is like adding 21 extra years onto a human's lifespan. So this is insane. So our results demonstrate that DAF12 was not required for the long lifespan of WDR5 mutants because removing the DAF12 activity does not affect the longevity of the WDR5 mutants themselves. And I'm still continuing to investigate this pathway in later generational double mutant strains, and I'm very curious to see what these results tell us. And I would just like to say thank you for this opportunity to present my research, as well as a special thank you to all the members of the Lee Lab.